Our final speaker in this session is Naftali Kaminski from the Yale School of Medicine, and he's going to be talking about lessons from the pulmonary fibrosis cell atlas.
So thanks for the invitation. I'll start by highlighting some of the people. Much of the work we did is basically a postdoc student who started in my lab, Tyler Adams. People have been asking me whether he's on faculty at Yale because he answers all the questions on the listservs. And he has been brilliant and done a lot of work, and then a few others that I'll mention during the talk. So basically, I'm a physician, and physician scientist. I study disease. The disease I focus is a disease called idiopathic pulmonary fibrosis. It's a chronic progressive form of interstitial lung disease. It is defined by exclusion of known causes of fibrosis. It has a very, very distinct, unique pathological presentation. There's around 200,000 patients in the US, 6 million worldwide. However, it's highly lethal. So 50% of the people will die within 2 to 3 years. So it's more lethal than most of the cancers many of us are afraid of. And actually, it accounts of 1% of the deaths in the UK over 60. There's 2 FDA-approved drugs, but they don't do much. And as many of us know, IPF is really the tip of the iceberg. Fibrosis is actually if our heart doesn't kill us, or the crazy shooter at the mall doesn't kill us, it is going to be fibrosis. So just to show you that we really have been aggressively, so this is basically publications in PubMed, normalized to 2010. This is all the publications in PubMed. They're driven a lot by cancer. This is pulmonary fibrosis. So we've quintupled the numbers of publications in many high-impact journals, and this reflects a completely renovation of the field, excitement, increase in funding, and a lot of basic science. What happens in pulmonary fibrosis? So we have a model. This is not really your usual inflammatory disease. There's recurring micro-injury that are driven probably by environmental injuries that are subclinical, that cause asymptomatic epithelial cell injury, epithelial and fibroblast activation, matrix remodeling, cell proliferation, and migration. And you sort of need to have some predisposing factors it's probably the most genetic associated complex disease. There's strong association with SNPs and the MAC5B promoter, some rare mutations in telomerase, so definitely an association with desynchronous aging, exposure to environmental factors, and lack of the involvement of traditional immunity or inflammation. My group sort of pioneered the application of high-throughput transcriptomic approaches to human lungs many years ago, and this is just an example of bulk RNA-seq. And bulk RNA-seq really helped us a lot. We identified significant novel insights, role of matrix metalloproteases, developmental pathways, microRNAs, and many others. And this is just an example of one review that looked at all the validated targets that came out of our work. But the thing is, look at this here. What we see here is actually tissue localization. It's a recurrent theme. And all of this is sort of hypothesis based on immunohistochemistry and other things. And as I'll show you later, actually, we know that one of my most famous targets, we actually called the wrong cell for it because we didn't have the data. And the reason is, and why is it so complicated, this is actually how pulmonary fibrosis looks. And I don't know how many of you look at histology, but if you see a tumor, there's a tumor infiltrating cell, and the tumor has an end and a beginning. If you look at an IPF lesion, per definition, you're supposed to have normal lung adjacent to this remodeled lung, and you have a myofibroblast foci, a few flattened epithelial cells, inflammatory response that's usually away from the area of injury. And what we were always interested to know is how come that this, beautifully designed, very simple structure of the lung for gas exchange. You have the type 1 cell that actually creates gas exchange. You have the pneumocyte type 2, endothelial cell fibroblasts. How does this become this mess? The other thing, of course, you look at this, you understand how complicated it is, that it's actually surprising that we've found anything meaningful in bulk data. So luckily, I was able to get my career before we had this realization. We try to address it by other ways. So this is a different approach in which we take human lungs after explant, we inflate them, we image them, we cut areas that are minimally affected, more affected, or severely affected. We can do we did bulk RNA-seq on those, and then applying computational models, mostly developed by Ziv Bar-Joseph groups, we could identify genes that are, again, this is weak, are induced early in the induction of the disease from normal tissue to minimally affected, and then moderate and later. However, we still could not address the sort of the cellular composition. So this was sort of the rationale for the IPF cell atlas. We needed to distinguish changes in gene expression from cellular infiltration. We needed to determine the presence of novel or atypical cell populations. We needed to look at cell communication at the cell level. And the main idea is to develop cell-specific therapies, cell-specific biomarkers, and potentially, actually, and in related to the previous talk, created a working model at the cell level of the fibrotic organ. Because if you had this model, now you could envision how you try to predict the impact of an intervention. If the model is calibrated, you inhibit X in fibroblasts, and you could model what happens to Z in a macrophage. The design was actually very simplistic. We said we'll have level 1 single-cell RNA-seq of several resources. One is dissociated lungs using a bank of tissue explants created by Van Rosses at Brigham Women's. And then analysis broncho lavage, which is basically washings of the lung that you could also use clinically with minimal risk. Analyze also peripheral blood and airway brushings. Level 2 is basically go back into the disease microenvironment using nuclear RNA-seq and analyzing these differentially affecting regions. So now you could have a measurement of the different cell types in location. Do spatial transcriptomics whenever we convince ourselves that we can get the resolution that we need, and do specific protein validations. Our considerations, basically numbers. We actually have enough numbers. We already did 100 lungs. We were not sure at the beginning, but the signal is so strong in the numbers of cells you actually get a really good signal. The other question is, what were the controls? And I'll be glad to speak about it in the end of the panel. When you're analyzing healthy, I would argue there's no such thing. So healthy is unborn, and that's probably not unhealthy also. Because the moment, especially the lung is exposed all the time to the environment. You walk behind a bus, you go out running, a change in the ozone levels, all of it is unhealthy. And in some ways, my argument is increased noise, increased diversity to actually be able to identify a disease signal. We wanted to be open-ended. I'll speak about data sharing maybe. The other challenge, of course, is rapidly evolving technology. You start a large study, but actually the technology changes in the middle, what you do with batches. And of course, these are precious samples. This is just a reminder of what we did. The first part, we take these lungs, which are dissociated, and basically we run the 10x platform. This is the numbers that we did. So we have around 350,000 cells from human lungs, around 160,000 cells from Yale, and we're getting there, and we'll have probably a million cells from everything by the end of the year. So the first lesson we learned is actually exactly what I told you. The dissociation matters, which is when we took dissociated lungs, what we saw is that the lungs were basically made of, that the lungs were full of macrophages. That's not true. The lung is not full of macrophages. What happens is that our dissociation method really favors macrophages. Now, you could actually calibrate your dissociation method for other things. And this is an example when we compare after calibration, single nuclear seq to RNA to single cell seq from in this case a mouse lung, we see that we move from a range of 50%, 60% of immune cells in the dissociated tissue to around 20% in the nuclear seq. I'm not saying that one is better. It's just that you need to know the preferences. And actually, you can actually calibrate it. You can improve the retrieval of endothelial cells, you lose other cells. You can improve the retrieval of epithelial cells. And I'm not a fan of the enriched methods because the more you deal with the cells after you got them out of the lung, the worse the signal is. But again, you want to know that every method has its trade-offs. But this is an example. Initially, we sort of were panicking, but actually, with enough tissues, we get a really good signal because the numbers are starting to get, and we're able to identify extremely rare cell populations. Like this is in epithelial cells in the lung, like neuroendocrine cells. That's a really a few hundred cells per lung. Ionocytes, although we're analyzing tissue parenchyma, there shouldn't be much airway there. We identify them. And also a population which is very interesting, which we call the IPF epithelial cells, which are very rare. But the cool thing, they don't show up in controls. But every IPF patient has like 60 of these cells, 60 out of 10,000. The data is amazing, and that's for sure. One is the images are beautiful. The second is the data is rich. Here you can see red is IPF, blue is control, COPD is sort of yellowish. You can see these are immune cells, and there's a chunk of macrophages in the middle. And indeed, those sort of macrophages that people have identified before really exist. That's highly reproducible. This is the strongest signal because we have a lot of macrophages. And again, we were able to reproduce the markers reported by Sasha Bisharin and also by Dvir Aron. So that's a real thing. Just turn on the clip. The problem is with our... could you turn on the animation? Sorry, where is it? No, we lost it. Yeah, this one. So basically, the other thing, we get hooked to these images which are two-dimensional, but they're three-dimensional. And we say, okay, these cells have SPP1 and marks MMP9. But actually, no. If you start looking three-dimensionally, there's some subpopulations that are fascinating, which suggests, again, that we probably don't need to do 1 million cells. We probably need to do 10 million cells. And then the continuity will be able to distinguish between different stages of differentiation for these different cells. Of course, I just put it because it's really nice. I'm gonna buy a 3D printer and basically make a lot of mobiles from these PAGA images. So the other thing we learned is very simple, but again, we couldn't do it before. The IPF lung is shifting. So for instance, here we can see that the IPF lung has, again, I'm not sure why I'm losing some of the animations, but basically you can see that the IPF lung has more B cells, less T cells. So of course, when people gave these patients steroids, they actually killed them. Now we can see the reason. They actually don't have a steroid-relevant response. They don't have a lot of T cell inflammation. On the other hand, there may be some things, and we're looking at these B cells, and we actually think that some drug reposition is going to be possible there. If you think about epithelial cells, and I mentioned to you the thing, so again, we can see these rare cell populations. We can see that type 1, the most important cell for respiration, actually disappears in the IPF lung. Now, you could say that this is only our dissociation, but no, this has been a repeating theme in other data. These cells are dying. They're probably the cell that's driving the process. And all of this crazy repair that's going on is because the lung is saying, oh no, my most important cell is going away. And then you try to compensate with type 2, and then with others, and then with fibroblasts. So that's what I think. And the other cell population, like ciliated cells, are coming in, and there's sort of this mystery disease epithelial cells we think may be important in disease. We have markers for them. Other cell populations are really different. Fibroblasts are completely different in the IPF lung, and also vascular epithelial cells, again, suggesting that some of the things may be primary and some of the things may be just responsive to the extracellular matrix. So, as I said, the data is amazing. Novel cell population, shift in cellular population, change in phenotypes of resident population, distinctive detailed characterizations of the aberrant cell-specific transcriptional programs is really possible, and it really gives us also a new appreciation of what is normal. Outliers really matter. So there's a tendency, if one sample looks crazy, to get rid of it. I suggest don't do it. So this is an example. This is now, we're annotating the cells based on the patients they came from. And you can see a lot of them are actually this. But you can see here, there's one population of fibroblasts, in this case, that comes from one, of macrophage that comes from one patient. I would argue that that's meaningful, and I'll show you an example in a second. In this case, we were looking at fibroblast population, and the same thing. We found one population that marked to one patient. However, Taylor and Jonas said, well, that does make sense. We have to find. We looked at the patient. This is a COPD patient who undergone transplant. He's much younger than all of our patients, like 30 years younger. And indeed, when we looked at this biopsy, he had a different type of emphysema. With evidence actually to external exposure, we were able to identify that he actually has a genetic induced emphysema, alpha-1 antitrypsin. But I'm going to tell you now, nobody with alpha-1 antitrypsin gets a lung transplant at the age of 30 if something weird didn't happen to them. So we're looking now at these fibroblasts, but this is an example of one patient could be extremely meaningful. Lesson 4, now you know what you did not know. So basically, that's a little bit about my career. I published this first microarray paper of human lung fibrosis in 2002, discovered that materializing MMP7 was involved in pulmonary fibrosis, put these beautiful stains, and really claimed that MMP7 was expressed in type 2 cells. And if you look, there's a million publications confirming this observation. We've also identified that MMP7 is a good peripheral blood biomarker. The only thing is, MMP7 in IPF does not come from type 2 cells. So it comes from different types of cells, including what we think are the disease-specific cells, but not really from type 2 cells. So again, we know that we're only as good as the best technology we've used in our careers. So luckily, I'm using better techniques now than I used 20 years ago. But the cool thing is that now we could look at all the biomarkers we've identified over the years, and we could say, this is coming from a type 2 cell. This is coming from an airway cell. This is coming from an angry T cell. And I think the impact of this, especially for environmental exposures, because you're not going to be able to biopsy patients. But if we have the map of the potential secreted molecules, and now we look at the blood, we could know what's going on in the lung in response to injury. And the other cool thing, the same type of data, now we take, I showed you that we did bulk RNA and identified genes that are induced early and late in pulmonary fibrosis, but now we can project the single cell data on this. We could project the single cell data on this and say, oh, this signal is actually mostly ciliated cells, and this signal is mostly type 1 cells, actually. So the loss of the cells is reflected in these trajectories. The last, I think it's the last lesson, is basically, it's sort of, again, as a history lesson, CGLSS is back, or avoid the temptation. So when I was, again, early on in my career, I'm a physician, so physicians usually want to name a medical condition. Now, I was never good enough to identify a new disease, but I was doing genomics, so I did identify a syndrome, which was common among people like me that did microarrays. And it was called the acute geneless stirring syndrome. And then it had a variant which was chronic, the chronic geneless stirring syndrome. Both conditions were characterized by sleep deprivation, I still have it, refusal to return to the bench, and excitement about funny gene names that I still have. The difference is it becomes dangerous if it's chronic because you may get paralysis of career. This was actually in a paper, so that's published. So the Kaminski tetrad is a thing. doesn't make my mother proud. Anyway, so that's exactly it. So I could sit and stare at this for hours. And not only this, I can waste time on generating these figures, right? But the main question is you want to think, where's the function? What do you learn from it? And another example is basically the gene list, right? So we can generate lists that characterize every cell, which are important. But if we stay there, as mentioned in the previous talk and others, we may have really nice papers, but we won't have an impact. So what I suggest is that we need to avoid AGLSS or CGLSS and basically make sure that we think about functions. So we need to look for niches. What are the cells that are close to each other, that talk to each other? The cellular circuitries, the temporal and regional interactions, because they're the most important. And this is just an example. This is a project in collaboration with Laura Nicholson with a brilliant student named Sam Murder. What we did is, and this was a conclusion of our IPF data, we said, oh, wow, we actually have these differences from normal, but we actually don't know what is normal. And then we said, maybe normal is the set of interactions that is conserved among species in the lung between different cells. And this is basically figure 1 of Sam's paper, trying to basically analyze rat, mouse, pig, and human lung, and identifying sort of ligand and receptor interactions that are conserved. This is, again, just a minor. Because of time, I won't go into this. But what you see is actually there are very strong, robust, conserved elements which really make sense, which actually highlight this concept that I told you, that a type 1 cell is probably the most important cell in the lung alveolus based on the reactions. And this is just a sort of a simplified version of all of these interactions. And the idea is, and we're replicating now, that once we have it, now we can look at the disease lung and see what is perturbed. And potentially things that fix this are going to be really better. So understanding the sort of these molecular circuitries that maintain cellular homeostasis in the organ, in distinct niches, may be the most important thing to actually lead to cure CGLSS. But maybe also IPF. So the lessons, I spoke about tissue handling, obvious, intuitive, but we tend to forget it. The one caveat I'm going to say, different people say that different methods are better. I'm going to say none. All methods are not good, and what you need is to triangulate and look from multiple ways. Second is, the data is really amazing. Outliers matter. They actually could teach you about biology. Now you know what you did not know, which is sort of cool. I mentioned CGLSS. And again, we're really excited the amount of discoveries. I've actually been thinking of doing almost like a lab shutdown for a few months, because what we need now is maybe to stop generating data and sit and think a little bit. We're not going to do it because we're, so again, I mentioned many of the people. We were lucky to get a generous donation from 3 Lake Partners to start this project. NIH and NHLBI is funding much of my research, including the newly funded normal lung aging cell atlas. And again, many collaborators. And again, I always mention the patients. This is where I come from. This is the advantage. So this is dedicated to the pulmonary fibrosis patients. If you are interested, you could follow the hashtags. This is a hashtag I started 2 years ago, Cure IPF, and the assumption that you could actually maybe even cure a disease, not only prolong it. So I'll stop here, and I'll be available for the panel discussion.